Hello, everyone. I am Kizzmekia Corbett, and I am the scientific lead for the coronavirus vaccine team at the National Institutes of Health in the Vaccine Research Center. I am going to tell you a little bit about everything you should know about the COVID-19 vaccines. So we all are aware that COVID-19 has caused many inequalities in healthcare. So the graph that you're seeing here shows the cases per 10,000, and you see that there is a disproportional amount of cases in minority communities like the Latino community and the Black community as compared to the white community. Those inequalities in the healthcare are basically based in our social and structural determinants of health. So what that means is that people of color are overrepresented in essential services and industries with increased exposure. There are often low-wage jobs that do not provide health insurance and the like. There is residential segregation, and people of color have increased amounts of chronic stress that render them more likely to get certain diseases. We are currently in our third wave of COVID-19 in the United States. This graph shows the three different waves, the first of which started last spring, and then another that started in early summer, and then the other that started in late fall right before the holidays. This graph shows the country in full. This graph shows the local community where I live in Maryland. So these waves are not just a countrywide problem, but also on a community level. It is estimated that we need to reach about 70% population immunity in order for us to have COVID-19 well enough controlled that we might be able to resume normal activities. Now, if we depended solely on an infection in order to reach that level of immunity, you could expect that about 40 million people would have to succumb to COVID-19 disease worldwide, which is completely unacceptable. So at this point, a vaccine is needed in order to prevent the dynamics of the pandemic.
A global race to find treatments for the sick and a vaccine to protect the healthy.
This ends with most or all of us being immune to this virus, and ideally that's through a vaccine.
Can solutions that usually take years
We are trying to move as fast as we possibly can
be found in a matter of months?
It is an extremely revolutionary approach to get into a phase 1 clinical trial 66 days after the sequence of a virus is released is a world record. A world record. But we don't really understand from a lay perspective what exactly a vaccine can do. Many people fully understand and grasp that there is a benefit to the individual. So a vaccine can reduce severity of illness. It can perhaps prevent infection. But what is lost to many people is the benefit to the community in all. So a vaccine has the ability to reduce transmission and make it safer to resume normal activities. With that, as I've said on Twitter, I believe that vaccination is community service. And the reason for that is because vaccination allows for us to safely and effectively create population or so-called herd immunity. Look at these stick figures in front of you. So there are two populations, the first of which is a non-immune population with blue people. The second population is an immune population or partially immune population where the green people cannot contract COVID-19. Letting the virus loose in that blue population, you can see that there's high transmission whereby the red people get infected with the virus. However, in the immune population, the green people cannot be infected if that virus is released, and so there is low transmission. And so you can see just in this picture how easy it is to disrupt the dynamics of viral spread by creating population immunity, essentially thinking of those green people as roadblocks to the viral spread. There are several ways to acquire population immunity via antibody responses, for example. The first of which is by natural infection. Now, we all understand that natural infection can potentially cause severe illness. People even die from COVID-19. So that is not the safest way to elicit immune responses. Also, we understand that the immune responses following natural infection are not as robust as what we've seen with vaccine responses. So the second way is vaccination. Taking all things into account, vaccines can be delivered in multiple different ways. The ways that you're hearing about with these new vaccines are gene-based delivery via messenger mRNA, and I will explain that later. But all in all, whether it be a traditional way of delivering a vaccine or in a more novel approach like we are using, the point of a vaccine is to elicit antibodies in a person so that a person's body can prevent them from being infected and also getting disease. There are also other ways to get antibody responses, like, for example, the direct administration of antibodies, and this is what you've seen with, for plasma therapy. And we will not be talking about that today, but those kinds of therapies, specifically by just giving antibodies to a person, that can be used before an infection or also after an infection. So it can be used almost like a treatment for the disease. Talking about this rapid development of the SARS-CoV-2 mRNA vaccine, the one thing that frightens a lot of people is that it just happened so fast, particularly more rapidly than we've ever seen any vaccine happen previously. But I wanted to just lay out why that was possible. So the timeline of development started really last January on the 10th, when the sequences of the new virus that was circulating in China were published online. Very quickly, we were able to go into a phase 1 clinical trial. So this is a clinical trial in about 50 people that just asked the question, is the vaccine safe? We were able to do that in 66 days in record time. And the reason why is because there was so much research that went into understanding how to make a really good vaccine for coronaviruses, even prior to this pandemic. So we studied vaccine development in the context of other coronaviruses that really helped to fuel this rapid development in the case of COVID-19 vaccines. How do we do that? This picture looks like a lot, but really just focusing in on that one particular spike protein in the middle. That is the protein that sits on the surface of the coronavirus. This protein's entire job is to latch on to a cell in your body and allow for the virus to get in. So one might hypothesize that if you can disrupt that interaction between the spike protein and your body cells, then you've created an effective way to prevent infection and disease. So that's why we want to target our immune response toward that spike protein using a vaccine. So we were able to study that protein in so much detail, able to see what that protein looked like, and able to gauge the type of shape we would want that protein to be in order to make a really good vaccine. And after assessing that for several years, we then had a really good vaccine insert or antigen. So this is the thing that antagonizes the immune system to create an immune response. So we wanted to give that spike protein as the vaccine antigen, but how do you deliver it? Remembering that if it's a pandemic, you have to move really quickly in order to beat out the virus like we're trying to do now. Making a traditional vaccine takes a little more time. So we wanted to use a system that would allow us to move very quickly, that was reliable enough and tested to uphold a manufacturability standard, and was universal enough in that we could plug in any protein that we might want. So we collaborated with Moderna, which is a biotech company that specializes in the delivery of therapeutics by messenger RNA. So messenger RNA is genetic material that basically sends your body a message to make something. In this case, we are using the message of the spike protein that we understood for the last 6 or 7 years. So basically, you are getting in your vaccine vial a message for the spike protein. This message goes into your muscle cells, and your muscle cells read that message, makes the spike protein, and your body senses that and starts to make a really good and specific immune response to it so that you're protected against the virus later. Now, because we understood how to do that, And because of our collaboration with Moderna that had been long-standing and very trusting, that we were able to, when those sequences of the virus came out, move in only 4 days with Moderna to make what's called GMP products. So this is the type of product that is good enough to go into a human muscle. They pulled the trigger or started to make the drug product in only 4 days and was able to ship that only 41 days later, which goes to show that if you can have a pure and safe vaccine ready that quickly, that the utility of the platform is really good in the case like now where we are trying to fight off a pandemic virus. Some of the steps that I'm skipping because of brevity of this presentation are all of the things that happened in the middle, where we were able to look at testing this vaccine in animals and assessing the protein in the vaccine to look at its shape and make sure that it was in its right conformation, and all of these things that helped to put together a large package of data on preliminary safety and immune responses that allowed us to submit to the FDA very quickly in order to get safe to proceed in order to go into our phase 1 clinical trial. And that's when the phase 1 clinical trial started. On March 16th of last year, almost a year ago. I will talk a little bit about the design of the phase 1, because the data from the phase 1 is data that allowed us to see that we were eliciting really good antibody responses in people, and also that the vaccine was preliminarily safe. In a small subset of people aged from 18 to 71, 3 different doses of the vaccine that has the brand name mRNA 1273 were tested. Just like how if you were to go get your vaccine today, these people in the phase 1 clinical trial got 2 doses of the vaccine at week 0 and week 4, and they have been tested for their safety and their immune responses ever since March of last year. So the data that you're going to see are specifically from the early on time points, but we have collected data since those doses early in last year. So this graph is very simple. What it shows is the amount of antibody responses in these people. And I've highlighted on this graph the different colors to show you the different age groups, partially because what the point of this graph is to show that is that just as in the green that you see on your screen, that in 100 micrograms, which is the dose that you get if you get the vaccine today, in people from the ages of 18 to 55, you get a really high antibody response. The same is true in people who are between or above the age of 56 and also above the age of 71. Looking at those blue and pink bars on the graph, what you see is that the people who are older have really good immune responses or antibody responses to this virus the same way that young people do. So this vaccine was able to make really good antibody responses in young people and also in older people. And it wasn't just at that really acute time point. So it wasn't just at, 2 weeks after your second dose. This is happening and it is staying steady for at least 119 days following the vaccine. So this preliminary data really helped us to see this vaccine is working and is eliciting really good antibody responses. And because of that, we're able to go into a phase 2 clinical trial and then go into a phase 3 clinical trial, which is a randomized placebo-controlled clinical trial where a large amount of people, in the case of Moderna's vaccine, 30,000 people total, 15,000 that got the vaccine, 15,000 that got the placebo. Those people went off into their daily lives after getting their 2 doses of either vaccine or placebo. If they got sick, they came back to the trial site and they got the question, what kind of symptoms do you have? And then they were either diagnosed by test with their trial physicians of having a COVID-19 disease or not. And after that, the cases were accrued over time. So in this particular example, there are 20 total people that got COVID-19 in the clinical trial. 2 were in the vaccine group and 18 were in the placebo group. So in this particular example, the vaccine had a 90% efficacy rate. So that is, if you got the vaccine, you had a 90% less chance of getting COVID-19 than people who did not get the vaccine. And that is how efficacy is calculated. And that is why it was really important for us to not just have a large amount of people in the phase 3 clinical trials, but to also have large amounts of people from various different backgrounds, really pointing to the idea of assessing safety and efficacy in people from all walks of life, whether they be Black, Hispanic, or Asian, and also people who had any comorbidities, such as, for example, 36% of the people in the phase 3 clinical trial had well-controlled diabetes. Looking at the data from the phase 3 clinical trial, these are firstly the efficacy data. So the purple bars shows the placebo group, and the orange bar shows the mRNA vaccine group. In this particular graph, what you can see is that as the cases over time accrue, so over time on the bottom axis, there are cases that are accruing in the placebo group, in the purple, in a stepwise fashion. Whereas in the orange, there are little cases. Actually, in this particular interim analysis, there are only 11 cases in the vaccine group. So that is out of 15,000 people. 11 of them got COVID, as opposed to 185 in the placebo group that got symptomatic COVID, and an additional 30 in the placebo group that got very severe COVID that required hospitalization. So it becomes very clear by the numbers that this vaccine is effective. Now, secondly, looking at safety. So the FDA terms these adverse events. In vaccinology, we call them reactogenicity. So this is, how does your body respond to a vaccine naturally, because the point of a vaccine is to stir up your immune system. So yes, after your first dose, many people experience pain or swelling or redness in their injection site. About 80% of people who got the vaccine, this is your body's way of saying, we know something foreign just went into your arm. Let's send some cells. Let's send some immune cells, and let's send some innate immunity over there to kind of help you to build up this long-lasting immunity that you want, specifically against COVID-19 spike protein. Then the second shot, there are more systematic events. So that is people sometimes experience one or more of headache or fever or fatigue. I personally, after my second shot, was fatigued and had a slight headache. So these things are normal in about 80% of the people who got the vaccine in the phase 3 clinical trial, and to be expected, but it is not an indication that the vaccine is unsafe. Actually, in contrary, what it is an indication that the vaccine is doing exactly what the vaccine should be doing, and that is basically shaking up or stirring up your immune system. Quickly, just to debunk some of the myths, as I know it is very hard to wrap your head around all of this very fast-paced data in this vaccine response, from which you're getting information from multiple different directions, particularly around a vaccine that is new. So this is a vaccine that is using technology that, while it has been tested in humans before and upholds some reliable manufacturability standard, it is new to the point of going into licensure. And so people are concerned about whether or not mRNA can affect your DNA or cause fertility effects. And those are just a hard stop no. That is untrue, that mRNA cannot affect your DNA, and it certainly will not affect your fertility. And in the phase 3 interim analysis, and actually since then, there have been no deaths that are specifically attributed to vaccines, mRNA 1273. So in closing, I want to just give you a brief outline of the whole globe's vaccine pipeline. So you hear a lot about the vaccines that are in the US, and I've just told you specifically about the one that's developed by Moderna, but there are over 200 vaccines in all, whether they be in preclinical testing or beyond, in this entire globe's vaccine pipeline. Many of them are in the preclinical stage, but there are 11 of which that are in the phase 3 clinical trial. So this is where the vaccines are tested in large-scale efficacy tests. So I talked about Moderna's vaccine. And then secondly is Pfizer's vaccine, which is similar to Moderna's in that it is also a spike protein given via mRNA. There's also Johnson & Johnson's vaccine candidate, which uses a spike protein being delivered in a very cool way. So they scoop out the guts, so to speak, or the replication machinery for a virus called adenovirus. So this is a virus that causes the common cold. So they use that to deliver the spike protein for the COVID-19 virus. Then there's AstraZeneca, which uses a similar technology as Johnson & Johnson, but just in a different vector or a different type of adenovirus. And then there's Novavax, which uses a protein-based vaccine. So this is a spike protein, not the whole virus at all. So some of the takeaway points is that none of these vaccines in this particular portfolio for the US are live virus. So you're not getting the full virus that causes COVID-19. You really are only exposing yourself to the spike protein. The other takeaway from this is that while I only show data from the Moderna vaccine candidate, there are major findings that are similar to that for all of the vaccines in the US's vaccine portfolio. And this is extremely important because as we think about the other vaccines, for example, Johnson & Johnson's or Novavax's, you want to be able to wrap your head around that the vetting and the criteria around assessing these vaccines were equally as stringent from not just the FDA perspective, but also around all of the data that was built up around these vaccines in animal models, for example, to show how really good immune responses these vaccines elicit. The efficacy of these vaccines is being reported almost daily, and especially as the phase 4, so to speak, rollout is happening. But from the phase 3 interim analysis, the efficacy for the Moderna and Pfizer candidates are above 90%, about 94%. AstraZeneca has at least 62% efficacy in the interim analysis, and Johnson & Johnson has about 80% efficacy in the interim analysis. And these two platforms have varied efficacy because they were tested not just in the US, but they were tested around the globe with various variants, etc. Thank you so much to the AMA for allowing me to be your guest speaker at your annual meeting. I wish everyone well. I understand that this pandemic is extremely stressful. It is the reason why we are coming together virtually. But as I like to say, just hold fast. And if you feel so inclined and feel informed enough, get your vaccines so that we can return to normal as soon as possible. I also did leave out my acknowledgement slides for brevity, but just a quick reminder that this work would not be possible without all hands on deck at the National Institutes of Health and the Vaccine Research Center and at Moderna, and with everyone all inclusively working together to make sure that this is a collaborative and effective and efficient effort.